Hi, let's talk about the endocrine system. So cells in a large multicellular animal need to communicate with each other. And there's two main ways that they can communicate with each other. They can communicate through action potentials, which is the nervous system, and they can communicate by chemically signaling each other. And sometimes these signals are local and are just signaling neighboring cells, and sometimes they're going into the bloodstream and signaling cells in distant parts of the body. But the cell has to be able to respond. The target cell has to have a receptor for that particular hormone in order to respond. And for the nervous system, the neuron has to be connected to or close to the target cell in order to have the response. So the endocrine system is hormones. These are generally going through the interstitial fluid into the bloodstream and then reaching their targets distantly in the body. The nervous system doesn't change where its signals go. The nervous system is hardwired with particular pathways and particular tissues being targeted by certain neurons. Although the endocrine system, which cells respond might change depending on receptor availability on those particular cells. And they often do the same thing or target the same tissues. There's some overlap between these, as you'll see when we talk about the nervous system in an upcoming chapter. So the type of communication that's occurring depends on what the actual signal is, what is the particular hormone or molecule that's being released by the cell, and how it's getting there. Is it going through the bloodstream? Is it going through just the interstitial fluid to neighboring cells? Is it a neuron that is communicating by an action potential? So endocrine signaling, generally what we're talking about is signals going through the bloodstream, and these are molecules that we call hormones. So they're being released by a gland, and they're going into the bloodstream and then reaching target cells, which could actually be close by, but they also could be very distant in the body. But that's what we call endocrine signaling. It's going through the bloodstream. It's a molecule that's being secreted by cells and diffusing into the bloodstream and going to distant parts of the body, or sometimes close things in the body. So there's also, though, sometimes signaling is not going into the bloodstream, and we don't call that endocrine. So endocrine signaling here are molecule, our cell in the gland is secreting a hormone, it's going into the bloodstream and then reaching target cells that way, although the target cells have to have the correct receptor. If they don't have a receptor to bind that hormone, they're not going to be able to respond to it. But we also have paracrine and autocrine signaling, which we saw already when we were talking about the immune system. There are cells in the immune system that secrete molecules that just target nearby or neighboring cells. They don't go into the bloodstream and travel through the whole body. They're not being secreted by a particular gland. Instead, they're intended to activate close by cells and get them to join in the immune response. And this happens both in the innate immune response and in the adaptive immune response. So we saw macrophages and dendritic cells, which will secrete and even the histamine secreted by mast cells is designed to target just nearby cells and recruit them into the immune response. We also saw examples of autocrine signaling. Remember, this root auto means self. So autocrine signaling is cells weirdly signaling to themselves. And this also happens in those same cells in the immune system. Cells that are paracrine signaling neighboring cells to join the immune response are often also have receptors for that very signal that they're releasing and responding to it themselves. So helper T cells, for example, if you remember, helper T cells, when they bind to the antigen that they specifically have a receptor for, they secrete molecules, interleukins, that bind to B cells and T cells and help stimulate that part of the immune response. So, and they also respond to their own signals. So helper T cells both are paracrine signaling to B cells and other T cells, and they're also autocrine signaling because they respond to their own signal and increase their response and multiply. So remember, cytokines are things that trigger cell multiplication as well. So, but we did see that already in the immune system. So we talked about those a bit already, and we will also talk about paracrine signaling when we talk about things like pain reception in the nervous system in future chapters. So, and that's going to involve prostaglandins, which are one of those paracrine signaling. So, paracrine and autocrine signaling play a part in a lot of important processes in the body, including pain reception, which we'll talk a bit about, also in reproductive organs. One of those signaling molecules that's used for paracrine signaling are prostaglandins. Prostaglandins are kind of a weird collection of molecules that are all very similar. They're made from fatty acids. They're modified fatty acids, and so they're mostly hydrophobic, but they have a little bit of an ability to interact with hydrophilic molecules. But prostaglandins are a stimulatory molecule. They are part of the contraction of the uterus. They stimulate muscle contraction in the uterus. They also make nerves that detect pain more likely to fire. So they increase the pain from inflammation is one of the things that they do, which is how ibuprofen works. Ibuprofen is the drug that's in things like Advil and Motrin are ibuprofen. And ibuprofen works on peripheral pain by blocking the release of prostaglandins. That's why it works so well. And that's why it's great for menstrual cramps as well, because it blocks that effect of prostaglandins in causing very intense uterine contractions, which is what menstrual cramps are. So that's why ibuprofen, that's how it works. It works by limiting prostaglandins, and it's great for mild to moderate peripheral pain, which is amplified by the secretion of prostaglandins. It's not, ibuprofen, as you probably know, isn't super effective for really intense pain like from a broken bone or something. But that's how it works. It works by lowering the level of pain. So we're going to talk about two different kinds of neuron signaling. One is synaptic signaling, and synaptic signaling means that a neuron is going to release a substance into the gap in between it and another cell, and that other cell could be another neuron, or it could be a muscle cell, and that's synaptic signaling, but it does involve the release of a molecule, so we will talk about that a little bit in this chapter. And we'll also talk about neuroendocrine signaling, and we see the word endocrine in there, and remember, endocrine means that something is released into the bloodstream, a hormone is released into the bloodstream. So neuroendocrine signaling means that instead of cells in a gland releasing this molecule, instead it's a neuron releasing molecules that are going to go into the bloodstream. And when we have neuroendocrine signaling, we're really talking about the hypothalamus. It's the only place in the body where that's happening. So we'll talk a little bit about synaptic communication. So synaptic, to have a synapse, you have to have at least one neuron. And the neuron could be, this is the axon, and we're going to talk more about structures of neurons in a later chapter. But a synapse, the synapse is the gap. The synapse is the gap between the neuron and the other cell. And that other cell could be another neuron, could be the dendrite of another neuron, or it could be a muscle cell, and there's a few other cells that they can communicate with as well. If we're talking about a neurosecretory cell, neuroendocrine signaling, that means that these molecules are, instead of going into binding to receptors in a neighboring cell, instead they're going into the bloodstream, and this is in the hypothalamus. That's where this is happening. So for the synapse connections to a muscle cell, so this is how your muscles contract. you have nerves, motor neurons that have, this is the axon of the motor neuron, and at the terminus of these axons, there is a small gap between the end of the neuron and the muscle cell. And there are receptors on the muscle cell for the molecule that's going to be released by those neurons. And for our muscle signaling, there are different kinds of neurotransmitters that can be released into these synapses, but the one that's being released in our muscle cell synapses is acetylcholine. It's a water-soluble molecule. It's released by exocytosis. So when a nerve impulse comes down the axon, an action potential, which we'll talk more about in the neuron chapter, the action potential comes down and triggers the release of acetylcholine. That in turn binds to receptors on the muscle cell, and that in turn triggers an action potential in the muscle cell. And that's going to lead to muscle contraction. There's going to be calcium release in the muscle cell, and that's going to trigger contraction. So that's how synapses work in a muscle cell. And for our neurohormones, things that are released by a neuron into the blood, and this is not going, some of them are going a long distance, some of them are going a very short distance. This is the hypothalamus that's doing this. And some of the hormones that it is producing this way are releasing factors that trigger the release of other hormones, like thyrotropin releasing hormone, oxytocin, gonadotropin releasing hormone, growth hormone releasing hormone, which is going to act on the pituitary. So in that case, it's not, the signal is not traveling that far. Vasopressin or ADH, corticotropin releasing hormone, which triggers the release of cortical hormones like cortisol. And somatostatin, which we're not going to talk that much about, but it does the opposite of growth hormone, basically. So you could think of it as the opposite of growth hormone. There's also external signaling, which we're not going to spend much time on, except just to know that it exists. And these external signals from one multicellular animal to another multicellular animal are called pheromones. And pheromones function in finding mates, in trails leading to food, like in the case of ants. But a pheromone is the same idea. Generally, pheromones are very small molecules, but they're organic molecules that are synthesized and released. This is a little picture here. This is, I forget the species of moth these are, but the female moths release a hormone to attract male moths for mating, and the males can be identified by these big fluffy feather-shaped antennae that they use for detecting the pheromone molecules from the females. Aquatic animals also have pheromones, but of course then they're not airborne, they're released into the water. There is some evidence that mammals as well, including humans, have pheromones, although this is a field of science that is, especially for human biology, has less evidence, but it's interesting anyway. So pheromones are outside the body. Pheromones are signaling other individuals of the same species. So there are some local regulators, things that are, again, paracrine signals in the body, like prostaglandins, which are modified fatty acids. Some are polypeptides, which some of those we already talked about in when we talked about the immune system. And one particularly interesting one is a gas. And this is also found in the bloodstream, and that's nitric oxide. And the discovery about 15 years ago, that nitric oxide was a regulator in the body was really surprising because it's not organic. I mean, NO, that's the chemical formula. It's one nitrogen and one oxygen. That in fact, it's a really important regulator of blood pressure, was discovered accidentally when an artificial blood was made to use in the field, in the military. Because one of the big problems they have when there's a sudden blood loss in battle is that the individual is too far away from potential sources of a blood transfusion. So they had this idea, well, we'll come up with a synthetic blood that doesn't need to be refrigerated, that we can take out into medics, can take out into the field and be ready to give to soldiers in the event that they need a blood transfusion. Well, when I started doing studies with this in hospital emergency rooms, because of course that's where they tested it, where are you going to find, people with gunshot wounds that you could practice on? Emergency rooms. So what they found was that when they gave people the artificial blood, their blood pressure would go up. And this was a mystery. They couldn't figure out what was in the artificial blood that was causing people's blood pressure to go dangerously high. And it was dose effect. So the more pints of artificial blood they got, the higher their blood pressure went. And it turned out it wasn't that there was something in the artificial blood, it's that there was something missing from the artificial blood. And the thing that was missing was nitric oxide, because nobody knew that it was doing anything in human blood. It was never looked for, detected, because it's a small molecule. Nobody was looking for it. So that's how it was discovered that nitric oxide is in fact an important regulator of quite a few different functions, not just blood pressure, but that it's important for nerve transmission. It's an important trigger of cell proliferation, the growth of new blood vessels, new capillaries. That's angiogenesis. It's an important part of the immune response. So it's a whole thing, and it's a very recent discovery. Hormones, so remember, hormone is something that's not signaling local cells like paracrine signaling. Instead, hormones are things that end up in the bloodstream and are going to distant places in the body. And we can group them into three big categories: polypeptides, which that would mean a protein, or if it's a short one, a short polypeptide, we would just call a peptide. So when you see that word peptide, that's just short for polypeptide. And insulin would be an example of a peptide hormone. There are steroids, which are all based on the cholesterol molecule. They're synthesized from cholesterol, and they are hydrophobic and not water soluble. And then amines, which because it has that word in there, amine, that means it's going to have amino groups on it. And amines are mostly water soluble. And the steroids are hydrophobic. So polypeptides, again, this means, if you remember back from last semester, a polypeptide or a peptide hormone means that it's synthesized at the ribosomes. It's made of amino acids, and some peptide hormones are fairly short. They might contain 40 or 50 amino acids, or up to 100 or so amino acids, depending. And insulin would be one of those. All the steroid hormones are synthesized from cholesterol, and those would include all the sex hormones, estrogen, testosterone, progesterone. Also, the corticosteroids like cortisol and corticosterone, and also, weirdly, vitamin D, which really shouldn't be called a vitamin. It's really a steroid hormone. And then we have amines, which most of them are water soluble, like epinephrine. These are synthesized. These aren't made by the ribosomes like polypeptides. They're not synthesized from cholesterol like the steroids. The amines are synthesized in a similar way that amino acids are synthesized. Thyroxine is kind of an unusual exception, that it's moderately hydrophobic, even though it falls into this category. So water-soluble hormones are secreted by exocytosis, and they bind to receptors on a cell surface. So, for example, epinephrine, when that is secreted by cells, that is going to bind to cell surface receptors. It's going to travel through the bloodstream, just being dissolved in the blood. Remember, blood is mostly water. And so things that are water soluble can just dissolve in the blood and travel to wherever the cells are that have receptors. Lipid-soluble hormones have a little bit more trouble getting around the body. They can travel across cell membranes. So when they end up in the bloodstream, they need to be transported. Lipids aren't just floating around in our bloodstream. That would not be good. Instead, they are all attached to transport proteins. And that's in fact what they're one of the types of lipid transport proteins that are found in your blood is what they're testing for when you get your cholesterol tested, which they really shouldn't call it a cholesterol test because they're not actually testing for the amount of cholesterol in your blood. They're testing for low density lipoproteins and high density lipoproteins, which are large spherical proteins, complexes of many, many proteins and fats that can transport phospholipids mostly around your body. So fat-soluble proteins or fat-soluble hormones have to do the same thing. They need to be escorted through the bloodstream because they're fat-soluble, and if they weren't, they would just diffuse through the walls of the blood vessel and not get to where they were supposed to go. Their receptors are in the cytoplasm or the nucleus of the cells that are their target cells. So not on the surface of the cells like the water-soluble hormones, but instead these hormones diffuse into cells and bind to a receptor in the cytoplasm, which will take it into the nucleus, or it goes directly into the nucleus and binds to a target protein in the nucleus. So water-soluble, just exocytosis, they're just going to travel into the bloodstream and then bind to a surface receptor. And this will trigger a response in the cytoplasm. Lipid-soluble hormones diffuse out of the cell that's making them, and they need to be attached to transport proteins in the bloodstream, which will allow them to reach their target cell. And the receptor is going to be either in the nucleus or in the cytoplasm of the target cell. So they're going to diffuse across the membrane. Cholesterol is present in membranes, and these cholesterol-based hormones can travel across membranes as well. They don't have a problem with the hydrophobic inner part of the membrane. So the signal transduction is going to lead to some kind of a response. If a cell has a receptor for a hormone, when enough hormones bind to enough of the receptors, that will trigger a response. And that's called signal transduction. And it's going to take place through the cytoplasm if it's a surface receptor. And this also amplifies the signal. So a single receptor can trigger the production of numerous activated molecules in the cytoplasm. So that's a way for signals to be amplified. And sometimes the response is a change in gene expression. Sometimes it activates enzymes that are already present in the cytoplasm by triggering them to become phosphorylated, for example, and become in their active form. There's many kinds of responses in the cell. Sometimes the response is for the cell to divide, to trigger mitosis. One hormone that's one of these water-soluble hormones is epinephrine, or you may know it as adrenaline. That is the kind of the British term for it. Epinephrine regulates many responses and triggers responses. It's released in response to stress, and that stress could be an external stress, and that response could be triggered by your brain, like you see a spider and you don't like spiders. That's going to trigger the release of epinephrine. And one of the places that epinephrine binds, it binds to muscle cells, for example, and lowers the threshold needed for that muscle cell to have an action potential, which is the jitteriness that you experience in response to epinephrine. But it also triggers a response in liver cells. So epinephrine binds to liver cells and will increase your blood glucose level by triggering the release of glucose, triggering the breakdown of glycogen and the release of glucose into the bloodstream, which for a fight or flight response, that kind of makes sense from an evolutionary point of view. If you're going to have to run or fight, some extra glucose in the bloodstream would probably be a good thing. So this is that response pathway. So here is epinephrine. It binds to a receptor on the surface of a liver cell. And G protein, that's something that you hear a lot in cell biology. There's a lot of G proteins. And the G refers to the fact that it uses GTP for energy and not ATP. That's why they're called G proteins. There's a lot of G proteins that do things like this in the cell. And one of the things that it triggers in liver cells is the production of what we call a second messenger. So here again is a way for the signal to be amplified in that cell. A single molecule of epinephrine binding might trigger the production of hundreds of cyclic AMP molecules. So cyclic AMP is a very common messenger in the cytoplasm that triggers responses. It can cause the activation of different enzymes, in this case protein kinase A. So a kinase triggers phosphorylation. So in this case, it's going to end up by triggering other enzymes. Protein kinase A stimulates other enzymes. It's going to promote the breakdown of glycogen, which remember, glycogen is how animal cells store glucose. So glycogen is made of glucose. It's 100% glucose linked together into these big networks. It's going to trigger glycogen to be broken down into glucose, and then it's going to be released into the bloodstream, and it's also going to inhibit new glucose from being synthesized or from being bound together into a large glycogen molecule. So these two things together are going to increase the amount of glucose in the bloodstream, which is a very well-known response to a stressful event, is to have blood glucose increase, which for somebody who's metabolically healthy wouldn't be significant or alarming, but in a diabetic, this response can trigger a very high blood glucose because glucose is not being responded to appropriately by the cells, either because they're insulin resistant or they don't have enough insulin in the case of a type 1 diabetic. So this is a little animation of that, what I just told you. So I'm not going to show it to you in here, but you can click on it in the PowerPoint and watch it. But sometimes it's nice to see an animation to help review things. So responses to the fat-soluble hormones, the steroid hormones, is usually a change in gene expression because they end up in the nucleus. That's usually where they're going. And something that's going into the nucleus, that, you know, that's where the DNA is. So, and gene expression, again, remember, gene expression means if a gene is being expressed, it means whatever that gene codes for is being made, which is either, sometimes it's an RNA is the final result, or it's a messenger RNA that's going out into the cytoplasm and it's going to make a protein. So it's one of those two things. But that's what generally what these steroid hormones are doing. So they bind to their receptor in the cytoplasm, and then they go into the nucleus and generally then bind directly to the regulatory region of one or more genes, target genes. And turn those on. And an example of one that's very well understood, because of course not, well, I should say for most genes out of our 25,000 genes, exactly how their expression is triggered isn't completely understood. But there's a few genes that we understand really well, exactly how they are regulated and what binds to the regulatory region that triggers expression of those genes. And one of them is this particular expression of a yolk protein being triggered by estrogen. Estradiol is one type of estrogen. We actually have some estradiol as well. But it binds to liver cells where yolk protein is made. And so here's some cute little frogs. Not the frogs that the research was done on. I'm sure that this research was done on the African clawed frog, Xenopus. And most of the time when you see research on frogs, that's the frog. But it's not as cute as those little glass frogs. So estradiol in the liver of birds and amphibians binds to a receptor in the cytoplasm. So and these organisms, they're not producing constant levels of estrogen. Estrogen goes up and down seasonally. So in the spring, when they're getting ready to lay eggs, estrogen increases, or estradiol, one form of estrogen, increases. It binds to a receptor in the cytoplasm. That goes into the nucleus and binds to the gene for this protein, vitellogenin, which is one of the components of yolk in frog eggs and in bird eggs. And so messenger RNA is now going to be made. That gene is going to be expressed. So this hormone triggers the expression of the vitellogenin gene. And that results in protein being made. And then it will be transported to the ovary to be deposited in the eggs. And this is a little animation of that same thing, which I'm not going to show you right now, but you can watch it on your own in the PowerPoint. There can be different responses to the exact same hormone in different cells. Sometimes there's a different shape or a different type of receptor. And sometimes the cells themselves are just different. They're not going to respond the same way to that particular hormone signal, like, for example, growth hormone. Human growth hormone circulates in the body, and many different types of cells in your body have receptors to human growth hormone. But a muscle cell isn't going to respond the same way as a bone cell, and it's not going to respond the same way as an intestinal cell. Those cells are going to respond differently, even though they may all have receptors for that same hormone. And epinephrine is an example of that as well. Liver cells are not going to respond the same way as a muscle cell. Your muscle cells respond to epinephrine by lowering the threshold needed to reach an action potential. Liver cells respond by releasing glucose into the bloodstream. So things that secrete these hormones are called endocrine glands, like the thyroid and parathyroid glands. Testes and ovaries are also both endocrine glands. So anything that secretes a hormone that's going to end up in the bloodstream is an endocrine gland. There are also exocrine glands, which we've already talked about some of those when we talked about the digestive system, like salivary glands. And interestingly, the pancreas is both an exocrine and an endocrine gland. It's an endocrine gland because it makes two hormones. It makes insulin and glucagon, which we already talked a little bit about. And it also makes digestive enzymes, which are going to be secreted into the small intestine. So the pancreas is a interesting and weird combination of different kinds of cells that have two very different functions in the body. Enzymes that are going to help with digestion and also glucose regulation in the blood. So here are where our endocrine glands are, and we're going to talk more about the pineal gland and the hypothalamus and the pituitary. The pituitary is sometimes called the master gland because it secretes stimulatory hormones that affect the other glands in the body. Hormones released from endocrine cells always travel through the bloodstream and interact with receptors. And that will trigger some kind of a response specific to that pathway. But there's also hormones that trigger the release of other hormones. We've already talked about a few examples of this, like endocrine cells. So this isn't coming from a specific gland, but in the small intestine, there are endocrine cells that secrete secretin. This in response to a low pH, so an acidic pH. So the small intestine, remember, the pH changes. We go from a very acidic stomach to a slightly basic small intestine. But this hormone is going to be triggered in response to a low pH, so a pH that's more acidic. And not a gland, but specific cells are going to secrete this hormone. The target cells are very close by in the pancreas, but it's still a hormone because it's going into the bloodstream. And it's going to trigger the pancreas to secrete molecules that are going to reverse that pH. So the stimulus, low pH in the small intestine, that's going to secrete specific cells, not a gland, but just cells in the small intestine. The S cells are going to say, you can tell they weren't being too creative with the name there. secrete a hormone called secretin. And that, although it's going to circulate throughout the body, the only cells that have the right receptor for that are in the pancreas. And specifically the exocrine part of the pancreas that's secreting digestive enzymes, and that's going to cause the release of bicarbonate into that fluid, which is going to inactivate or react with the hydrochloric acid that is what's making it acidic and neutralize it. So it's going to neutralize the acidic nature of the stomach chyme that's coming out into the small intestine. So, and this is what we call a negative feedback loop, because releasing this is going to raise the pH, and that's going to lower the signal to the endocrine cell. So as the pH then is returned to the level that the small intestine prefers, the signal is going to decrease. The amount of secretin produced will be decreased. The pancreatic cells won't have as much signal to respond to, and bicarbonate release will decrease. So this is this kind of negative feedback for a response is normal. We already talked about some negative feedback loops with maintaining homeostasis for body temperature, for example. This is a little video on regulating blood sugar. I'm going to play it in here just because it's a nice explanation. It's similar to the video that you already saw in a previous chapter, but this is kind of an important topic, so we'll watch this one.
The sugary snack you devour enters your digestive system and is broken down to simple sugars like glucose. Glucose enters the bloodstream, causing an increase in blood glucose levels. But various mechanisms bring blood glucose back down to its normal level, the set point. This is an example of homeostasis, the body's tendency to maintain relatively constant internal conditions. Hormones produced by the pancreas regulate blood glucose levels. Let's zoom in. When blood glucose levels are high, glucose molecules leave the blood and enter beta cells in the pancreas. The beta cells respond by releasing the hormone insulin. Insulin enters the bloodstream and is transported to cells all over the body. Let's see what happens in the liver. Insulin binds to receptors on liver cells. This causes the cells to take in more glucose. Inside the liver cells, glucose is converted to glycogen, a storage molecule. Blood glucose levels decrease as glucose is taken up by liver cells and other body cells. As a result, less and less insulin is released by the pancreas, and blood glucose levels stabilize at their set point. This is an example of how negative feedback maintains homeostasis. What happens if you've skipped lunch and your blood glucose levels are low? Let's zoom into the pancreas again. When blood glucose levels are low, alpha cells in the pancreas release the hormone glucagon. Glucagon enters the bloodstream and acts on target cells in the liver. Glucagon binds to receptors on the liver cells, signaling the liver cells to break glycogen down to glucose. Glucose is released. And blood glucose levels increase. As a result, less glucagon is released by the pancreas, and blood glucose levels stabilize at their set point. In this way, two hormones with opposing effects allow the body to maintain homeostasis of blood glucose levels. In diabetes, the body is unable to maintain homeostasis of blood glucose levels. In type 1 diabetes, the beta cells of the pancreas are destroyed by the immune system, and so no insulin is produced. In type 2 diabetes, the pancreas produces insulin, but target cells do not take up glucose. In both types of diabetes, when blood glucose levels rise, cells do not take up the additional glucose, and so blood glucose remains high. This example shows the importance of homeostasis in everyone's life.
Let me get my pointer back here. So in a simple neuroendocrine pathway, so neuroendocrine, remember, this is when a neuron is secreting a hormone into the bloodstream, and we're pretty much talking about the hypothalamus here. So a neurosecretory cell, where that is a neuron that is secreting a hormone into the bloodstream to travel to the target cells. Sometimes it's not going very far, sometimes it's going very far. And one example of that would be oxytocin. So oxytocin is necessary for milk letdown in mammals. So mammary glands make milk, but there's also muscles present in the mammary tissue, smooth muscle, involuntary muscle, that will start to push that milk out of the mammary tissue in response to the infant suckling, and that's where oxytocin comes in. So, and this is an example, an unusual example of a positive feedback loop, because most of the time we have a negative feedback. This one is a positive feedback. Okay, so the initial stimulus is the suckling of the infant. This, because this is a sensory input, it's going to the brain. And in the hypothalamus then, this triggers the release of the hormone oxytocin. It goes into the bloodstream. So here you notice that the cell body of these neurosecretory cells are in the hypothalamus, but they extend their axons into the posterior pituitary. So the posterior pituitary gland is really part of the brain. It's part of the hypothalamus. It's an extension of the hypothalamus. That's the posterior pituitary. So these neurons extend into the posterior pituitary. They secrete the hormone oxytocin into the bloodstream. The cells that respond are those smooth muscle cells in the mammary glands that are going to squeeze the milk out. And when that occurs, that response is called letdown, milk letdown. And the milk starts to be released, and that's going to trigger the infant to increase their suckling. And this initial, the milk letdown takes a few seconds, maybe 10 seconds. And then the milk is released, the infant increases the intensity of their suckling, and that triggers more oxytocin to be released. And it's going to reach a limit. There's a maximum amount that these smooth muscles can contract and squeeze out the milk. But this is a rare example of a positive feedback loop. And without the proper stimulus, milk letdown is going to be decreased. So that's an example of positive feedback. Most of the time what we have is negative feedback, and most examples that we would have in homeostasis and hormone production, usually the response triggers a reduction in whatever the initial action was. That would be a negative feedback loop. A positive feedback loop means that the response will increase as the initial stimulus increases, the response increases, and the stimulus increases. That's a positive feedback loop. And there's many fewer examples of that. Most of the time it's negative feedback. Okay, so let's stop here for this video, and in the next video we will talk about specific examples of different hormones in the body and what they do.